All right, so we have 30 minutes for this panel discussion. And at any time, audience members are welcome to stand up and bring us questions. The idea of the panel is to really kind of summarize the overall theme and the kind of take-home messages from today's talks. I'm going to get us started with the first question. So some of you are more deeply rooted in natural history specimen collections, while others are primarily using living or even art collections. I'm wondering if you can speak to the importance of utilizing more than one collection type in the research that you do. And that's for anybody.
I'm a little bit out of breath, but I can start. So when I first started trying to look at herbivory on herbarium specimens, I did so using digitized specimens, and I noticed a few things. One was that using just the digitized specimens, I wasn't going to be able to get a handle on whether the specimen was eaten outdoors or indoors without really having that experience with the physical specimens. Catch my breath. I also noticed that some of them were not displayed digitally because they were rare or endangered. And I noticed that the damage was just hard to deal with as a beginner without actually seeing the specimens. And then I was able to use the physical collection, collect the data, and then now we use digital collections quite widely because I'm working with an expert on herbivory and using artifacts in this way. So I think whether I use digital or physical specimens depends on a lot of different aspects of the work, including expertise level. So they're both really important.
I would add to that I think the ability to have the multitude of different collections really gives you a depth that we can't get always. And it also is conserving the natural areas where it may be harder and more inaccessible. And so to travel to these places, if we actually can have both herbarium specimens, we can have seeds and seed banks, which may not always be collected in herbarium specimens, and then we can have living plants. And I think that being able to have access to all of those kinds of collections means that we can learn so much more and we can do so much more research in a meaningful way and really push forward. The living plants means we can do things like cryo and learning how to do tissue culture. And the seeds allow us to be able to, really test long-term viability and long-term success of storage. So I think I'm for as many kinds of collections as possible.
Awesome. So I love Lucia's reference to taxonomy being a team sport. I really feel like there's a t-shirt in our future with that. So yeah, love that so much. And really collaboration was a strong theme in many of your presentations. I'm wondering if you can give an example of a scientific advance that you have made through collaboration that absolutely could not have taken place had you not been collaborating with other collections-based researchers and institutions. And it might just be everything you do. I don't know, but maybe there's some good examples of those collaborative projects.
Well, I think just one important way in which collaboration is important is international projects, because if you're working with people in other countries, you often really need their expertise in the languages and the institutions involved. I think the days are gone where researchers can just sort of fly to another country and collect specimens and take materials. So the countries won't allow it to take place, but also if you have collaborators locally, you have a much richer experience and you understand the species much better.
Well, yeah, I'm trying to think. It's hard for me to think of any of my projects that were not collaborative. Yeah. But maybe I'll highlight one project that we did in the Amazon basin where we actually work closely with geologists. So we wrote the proposal together, we thought about the project together, and it just changed my whole perspective on everything. And at the beginning, we couldn't even talk to each other because I didn't realize how difficult some of the geological literature was. And it was so completely super insightful. Yeah.
Awesome. There was also a wonderful sort of hidden information theme to many of the presentations today. And I'd love to hear about the most surprising thing that you've discovered using collections. Most surprising.
well, you were gonna like this, mites. So we did a project with mite domatia, and then sometimes we would find the mites in the domatia. So we started just looking at the morphology and so forth, and then after measuring a bunch of specimens, suddenly there they were. So it was really interesting because we had some hypotheses about how the mites would be, and we actually found them.
Can you explain for the audience what domatia are? I mean,
Oh,
they're
yeah.
fascinating.
So, so this is in the Bignoniaceae. There are different types of domatia, but on the axils of the veins, there's a little, like a little package actually that are, sometimes they have little hairs or sometimes just a little, the veins are a little longer. And there's some mites that actually use that as housing. And they clear some of the leaves, and so they help protect the leaves from herbivory. And we started just doing a project to see how the mite in domatia evolves, and we were just measuring a bunch of different mite domatia throughout a phylogeny and looking at the evolutionary patterns, and found the mites themselves.
You're right, I do love it. I've heard of people doing some of these same things with ants, maybe thinking about looking at domatia and things. The ants are often preserved within them. It's so cool. I was not expecting for herbivory to increase in plants. I was not expecting that. I was expecting to have really clean hypotheses, and herbivories didn't decline with insect decline. And so I think that herbaria have like transformed the way that I look at my own research simply by showing that pattern over time, much like what Richard saw in plant phenology. Some of these patterns, because the data are so long and go back so far, you can really see these long-term patterns that show up more strikingly than I would have imagined, and the herbivory is one of them.
I have a little example. It's from entomology, but there was a woman on the Funk list, Sophie Letterlow, who was known to be, she was hired as the first female elevator operator here at the museum, and eventually became a scientific assistant in entomology. But we couldn't find any specimens that either she collected or identified. But looking through Smithsonian annual reports, we did find some mentions of her work. And one of them had said that she had remounted or rehoused 300,000 tick specimens in a year. And it was just like, oh my gosh, like that's such a crucial, amount work in the collection, but we really wouldn't have found it otherwise. So I just think there's so many things that we're just not even finding yet, but having these associated documents and archives digitized really just provides so much more information.
Oh, can I talk about a surprising specimen?
Yes.
So I was at the Harvard University herbaria on like a Saturday, as one does during a postdoc, and by myself, and I opened up this specimen of Bacteria tinctoria. Does anybody know what the common name for that is? Some sort of indigo, I think. Yeah. So it was the specimen had a attached newspaper article that talked about how there was a family that was foraging, and they thought it was wild asparagus, I think, and they ate it. And so I don't remember if the article said they all died or if just a few of them died, but there was this plant that I was just trying to look at insect marks on, and suddenly learned that the reason why it was collected was like a medical issue, and they'd brought it to the herbarium for that. So I do think that there is a lot of human history that is hidden in these collections.
Absolutely.
Just want to mention that starting when we became interested in climate change research, we just started searching for old historical records which might be relevant to climate change. And it's been my experience, both in the United States and in other countries, is that if you start looking for old historical records and journals and information, you often find something. It might not even be what you're looking at, and that's actually an important principle, is that you start looking for something, but then you might discover something else that's completely different. In our case, when we were starting to search for records, and just someone, a friend of a friend, told us about Thoreau's records, And as soon as we saw those records, we knew that they would be amazing. But I've had similar situations in other countries where you start looking and you have to make that effort to start finding things, looking for things, and then you often make amazing discoveries.
Absolutely. I will just say, audience members, you are welcome to ask questions. I have a whole list of them myself. It sounds like Gary might have some from our virtual audience.
Yeah, a whole bunch are coming in virtually at least. So this one question is about citizen science, and actually it's about citizen collectors. Are there organizations on a local or regional level that utilize citizen collectors for submission to herbaria? So are there herbaria that actually accept citizen collections?
I'm no curator, but my impression, at least in the United States, is that collections are being very judicious about what they accept because it costs money to store specimens and to curate them. But when I visited the National Herbarium in Japan, I saw this really surprising pattern that's definitely not in the US, where collections have stayed steady over the last 100 years. There are just as many collections being submitted now as there ever were, despite decreasing taxonomists. And I was like, what is happening here? And the person who was hosting me said, oh, we accept citizen collections. So there are a lot of people in Japan and outside Tokyo that collect for fun, and they can accession their specimens. So I assume in some places that's allowed, but I don't know of a specific organization that collates and does the work of organizing.
Yeah, there's quite a few university herbaria that have volunteers that are associated with them that they do collecting. So they're not really just from the public generally, but you can become associated as a volunteer with different organizations.
Yeah, and I was going to mention in Brazil too, we would often have different people who collected for fun. But if the specimens had the appropriate information, was something interesting, we would always include in the collection. Yeah.
Okay, another question actually ties in botanical gardens and herbaria, and that is collecting specimens at botanical gardens. And it says, among the many values of botanical garden collections is that they grow under known conditions over many years, allowing researchers to study changes in phenology, herbivory, anatomy, reproduction, etc. over those years. Some of this might be captured in voucher herbarium specimens. Do any of you know of researchers taking advantage of this?
So using cultivated specimens for, scientific research.
We certainly have used herbarium specimens over time to look at phenology, but you can also use them to look at other characteristics, for example, stomate anatomy, leaf morphology, and also sometimes physiological characteristics. So actually by looking at isotope ratios, you can use those to look at water use efficiency, which might be related to weather conditions. So there's a lot of things that you can do with herbarium specimens collected over time at botanical gardens.
Yeah, I'll just add quickly that there is a collection at UC Davis called the Oak Grove and the Shields Oak Grove, and it's a collection of trees, oak trees, something like 200 out of 500 global species have been planted there, all sort of at the same time. And it's been studied as a common garden over time. And I do have a student, Mariel Hansel-Friedman, who's using those to look at herbivory. The last time the herbarium specimens were collected from there, which was in 2000 versus more modern collections that we did in more recent years. I wish I could think of someone else. I'm sure there are other examples.
I have more online, but if there's any in the audience, feel free to, come on up. While he makes his way to the microphone, I will ask one more. Thanks for the great talks. Regarding the curation of collections, is there anything that would make your work from herbarium specimens easier or more impactful in terms of curating the collections? Something that would make it easier or more impactful?
I can say something to not do. So I worry sometimes when I give this talk because sometimes people come up and say, oh, I'm going to collect specimens with herbivory now. And it's like, no. The collection methods have to stay consistent or else we get erroneous patterns in the data. So I would say, sticking to traditional collection methods so that the methods are as consistent over time for lots of different kinds of climate science is useful. And also just digitize, digitize, digitize.
Yeah, great talks. Thanks to all the speakers. I was wondering if you like go on GBIF and you look at a trend line of collections over time, right? I think we've all seen that it kind of peaks in the 70s and 80s, and then there's like this slow, continuous crash until we get to the present day. And I guess I was really curious as to what you just said about that line being stable in Japan because of, and I guess I'm just thinking in terms of our collections and big collections all over the world, you know, I think that trend line is relatively, maybe Japan is the exception. But like, yeah, if we were to extrapolate out 100 years, you know, or more, like, do we think that we expect that pattern to repeat where like we get this peak and then a crash and then a peak and then a crash? Or is there a way to make it sustainable, you know, maybe where there's some kind of flat line? Yeah, just thoughts for. Or maybe why did it crash if the panel has any thoughts? I understand we're all overwhelmed with how many collections we have right now, but I guess as a early career scientist who has used a lot of collections for both morphology, ecology, genetic work, and I've been really, I got DNA out of a specimen, a Pringle specimen from 1881 that I was able to use in a phylogeny. So it's not that you can't get them from old specimens, but the quality of the data and my ability to integrate it with other data sets is limited by age in many. So I'm thinking of the graduate students in 60 years.
I feel like Lucia might have more nuanced thoughts about this, but it seems to me that maybe the funding for these sorts of things is being cut, and expeditions, that sort of thing, funding is being cut. And so maybe writing the collection of specimens into fellowships and grants might be a nice way for postdocs and graduate students, trainees in general, to contribute to the collections and make sure that they are at least plugging along and we get the types of data that they're interested in. And I think another thing from my research that I am selfishly excited about is going back to places where there is a nice history of collecting and then continuing those efforts so that we can continue longitudinal monitoring in various ways.
Just like to also say that one of the main purposes of collections was to look at the distribution and abundance of species. And we're actually able to sort of build on that using iNaturalist data. So iNaturalist data is increasingly being used to look at how species are changing or where they're found and not found. So I think that it would be great to have more herbarium specimens, but we're also having this other resource now of iNaturalist records, which provides a very good modern kind of extension of that existing historical record. We just have to preserve the historical record.
Thank you very much. Or Lucia.
Yeah, no, I'm just thinking here also in my mind, like what are the reasons for the pattern, right? And there's multiple, right? Also, we have so much to do, right? So in the past, people would collect, would describe, and now we need to do the molecular work. There's so many other things that we do. And then in the end, I'm thinking a lot about a lot of the graduate students that don't have as much time to go into the field. When I was an undergraduate, we would be all the time out in the field, and graduate school too, but it just feels less time. And at the same time, the whole thing with permits became so complicated that it adds another layer, which is so unfortunate. But in some ways, going back to the collaborations and making sure we are always doing the exchanges and collecting multiple specimens for multiple collections. Somebody said of not putting all the eggs in one basket. I think it was you, and I could, yeah, really relate to that, and make sure we are always exchanging those specimens. And if there's one thing that is so really urgent, is collecting, right? That's the one thing we cannot do 30 years from now. So we all need to find time for that.
Thank you so much. Appreciate it.
Thank you. We've got another question from the audience here.
Okay, thanks. So I agree with everything that the panel have said already. I think it's also going to be a matter of time before we can really deduce trends in collecting globally, because until the regional herbaria, which are still very actively soliciting collections and sending people out to collect, until those herbaria are thoroughly digitized, I don't think we're going to get a particularly accurate picture. I should also say that there are lots of places in the world, particularly where there is high biodiversity, where citizen scientists in particular are contributing a lot to collecting, because they, in some cases, are better resourced than the scientists themselves, and where the challenge is greatest. And so it's a matter of all hands on deck. And I know that that's already been mentioned. But one of the questions to the panel is, as we get more out of these digital collections, have any of you arrived at a sort of how you're thinking about how frequently we should be collecting so that that longitudinal record is maintained? And particularly given that there's evidence in certain places that collecting rates are declining, and are we facing the possibility of having gappy data because we're not collecting or recollecting frequently enough? Thank you.
Yeah, I think Lucia spoke to that a little bit, but there's other things that cause gaps in data in collecting as well. So maybe we can speak to some of those other things that might cause those gaps.
Yeah, and I'm also thinking about those projects in the past, like the Brazil Flora project, and then lots of collecting in those times. We so need more of those, and making sure we can continue to have the continuous record.
Yeah, it's interesting. I mean, the collections that we have were not made necessarily for ecology. And so I think for a lot of species, we don't have much of a longitudinal record, or at least it's very curvy in terms of how many specimens of particular species are out there. And so as an ecologist, I'm perfectly fine with like finding the richest collections or the collections that are appropriate for my uses. But I guess the issue of where to collect is one that depends on so many different interests. Like even on this panel, we have so many different interests in collections. And so I think it's a really complex thing that probably deserves a lot of discussion to be able to honor everybody's interests, and then also the potential unknown future interests of scientists.
Harry's got a question from
So I got another question online, this one from James. It's about digitization. Are any of you aware of any efforts to expand and organize images of micromorphological characters of specimens to accompany digitized specimens? This would be a fantastic and much needed addition to digitized collections, particularly for groups with cryptic characters such as Cyperaceae and Xeridaceae.
Oh, I love this, and it kind of really speaks to the Domatia you were talking about earlier too.
This is the idea of the extended specimen, right? Like the fact that you have these physical records and then you have the metadata associated with them, like who collected them, where it was, what year it was, what day. And then there are all of these other data sets that are then derived, like is the plant flowering or not? Did it just leaf out? Does it have insect damage of various kinds? Has someone DNA sequenced it? Has someone sequenced the fungi that live in the leaves? All of these are data that are extensions of the original specimen. And there have been a lot of people who think deeply about the extended specimen, and I am not one of them. So if anybody has any experience with that.
I was just going to say that with the morphology, I always hoped that Morphobank was going to be that place, but somehow it didn't really quite touch.
Well, I mean, possible extensions are, for example, using artificial intelligence to take the image and get a lot of characteristics of the leaves on them and the stems and the flowers to make a much more detailed description. But another possibility, which actually Lucia mentioned, was hyperspectral imaging. So this is a technique where you're shining a beam of energy at the leaf under a variety of spectra and then seeing what is reflected back. And this gives you extremely detailed information about the leaf surface and the hairs and cell types, and also information about the different elements in the leaves and different isotopes and ratios of them. So you can actually get a huge amount of information potentially just by putting a beam on the leaf just for a short period of time. And this is what is a really exciting development in plant biology, because you'll have all this vast amount of data which is associated with the herbarium specimen, which might be generated at the same time that it's being digitized.
I'll also add that in terms of, in terms of the extended specimen, I do remember reading papers from Pam Soltis, who's in charge of IDIG Bio. So I do think these are the discussions that are being had at the highest level of people who organize digitized and physical specimens for the United States is how do we take all of these resources and put them together?
I would add this is not for the herbarium specimens, but it is a way that we could tie in other databases that are digital. And this includes some incredible work that is being done across seed banks, and they are digitizing and categorizing and imaging all of the different seeds. And there's some really exciting stuff coming out of Lion Arboretum in Hawaii, where they're creating an AI assisted program to be able to really image out and help ID the species that they're training for the seeds. And so seeds are, obviously I have a very strong passion and background in seeds, but this is something that could really be tied into any of the herbarium specimens where you could actually link a seed, these seed databases to your herbarium databases. And so these derivative collections, I think, are really important to then link into our herbarium collections as well, because that's where you get that depth of information. And there's a lot of gardens out there that are trying to do derivatives. We have a derivative project that was, collecting tissue, herbarium vouchers, seeds, you name it, mycorrhizae, if we have it, into collection, tissue culture for all of our collection so that we have that full depth. And then you can put it, attach that to your herbarium specimens as well so that you know what's linked together. So.
Yeah, I love that integration of natural history to sort of more traditional collections and living collections, and the sharing of information between those. It really makes it a robust approach. We have a question from the audience. Ben?
So I have a sort of unanswerable question, but since we have a bunch of experts on the stage, there's been a number of different types of collections discussed, herbaria and living collections and seed banks and germplasm and DNA. Obviously, with different goals and different skill sets, you have different foci, but do you each have a perspective on, given also there's been reference to the constraints, limited resources and abilities, what is the most important things to focus on? Where should priorities be? And just as a maybe easier secondary part of that, are there efforts, like was mentioned with Botanic Gardens and meta collections, to go beyond the living plant collection, but on these other types of collections to coordinate among institutions and focus across collections where to have the most important things done?
I will argue that probably seed banks are one of your most economically, both for space and the amount of genetic diversity and the number of collections that you can hold in one space. So we often prioritize seed banking, but then there's of course all of the species that can't be traditionally seed banked. And I think that living collections are very important for getting that people engaged with living plants. But I think that there's then the need for herbaria to be able to be accessible. And I mean, it's part of why I think that it's hard, you can't pick your favorite child. I think financially, I would pick always the seed bank because it's just about genetic diversity for us. And I don't remember your other question. I'm sorry.
Do you know of efforts or conceive of efforts to collaborate beyond just, ex situ collections, but to include herbaria, and so that different institutions are focusing on their strengths, but in a coordinated fashion? So you're thinking about herbarium specimens as well as live plants, as well as seeds and DNA and whatever the components are.
I would argue that the Center for Plant Conservation and Botanical Gardens Conservation International are trying to do that with their different national collections and tying together gardens and their the database, the plant search database, to start to get people to report and keep better records of all of these different materials so that there's more open access to that. So I think there's some interest. And I think that that's where working together in collaboration, that's really most important.
I mean, I will also add that one of the most exciting opportunities is with all these citizen science networks like iNaturalist, eBird, Project Budburst. I mean, that's what people are doing. And so if we want to involve the public and increase interest, that's the way to do it. I find with my students that I can hand them field guides and they just look at it like, what is a field guide? What is a book? But if you tell them to start using iNaturalist on their smartphone and start taking pictures with their smartphones, they're very enthusiastic. So I think we have to go with what students and the public are interested in. And it's also a great resource in terms of botany and natural history.
We've got one more question from the audience over here.
Hi, thank you for all of your wonderful talks. I had a question about the global initiative 30 by 30, which aims to put 30% of land into conservation by 2030. I know that the idea is that we can save half of biodiversity by doing that, but there's no real consensus about how much biodiversity that we're saving until we have the inventories in place. And I imagine that a lot of these collections are the basis of claiming where the biodiversity is the highest, what are the priorities for land conservation, and in general beyond 30 by 30. So I'm wondering if any of your collections or projects have been touched by 30 by 30, or if you've been sort of the hub of these things. What is the role of these collections in like conservation efforts globally?
So I will give you obviously my biased perspective. I think that with the 30 by 30, there's a lot being done to tie in these KBAs, our key biodiversity areas, and the data that is often pulled from that includes on the ground surveys, but I do believe that it also includes herbarium specimens. And as those key biodiversity areas are identified, it's really about getting as much data into those KBA, this KBA database, so that we can actually start to prioritize. And so that includes any data whatsoever. And I think that that is where trying to find these more global databases like GBIF that can be pulled into that information and making sure that we're sharing our data as much as possible. I work with a lot of poached plants as well, so I absolutely understand the stresses and pressures around sharing lat long, because it's a very serious issue. But I think that being able to pull in that data safely is critical. And of course, GBIF and any of the, herbarium data is really important, as well as on the ground. And I think that's also where we have to be including our communities, our local communities near these biodiversity areas, and get them engaged to help, because the local communities often know about, when you start talking to them and you go in the field with them, they tell you, oh yeah, I know a plant that looks something like that. Let me take you to it. And so getting that knowledge and getting it into databases, I think is a huge challenge, but really important because if we're just drawing lines on the map for the 30 by 30 that don't actually have data behind them, we're not making, it's just paper to pen and it doesn't actually do anything. But I think it should. I'm all for it.
So as always happens with this panel, we are past time. And so I'm going to wrap up the panel by asking each of our panelists in the spirit of the final word and the radio show, wait, don't tell me, what is the next big thing in your collections-based research? I'm going to start with Rebecca.
The next big thing, oh my gosh. I really think it is that sort of overall knowledge graph or like cross collection search where we are just not only looking at any one collection or herbarium, but we're really looking across them at aggregated data and also including, other disciplines in there too. So getting the library, getting information from full text in the archives and really being able to like create a network and a map of what people do. You can do it piece by piece now, but being able to do that at scale, I think is really exciting.
I mean, I would say the next big thing is using herbarium specimens and historical floras, which have been used to map where species are and what species are in different places, and combine these with, for example, iNaturalist records or records, for example, comparable records for birds using eBird data, and really using these kind of modern citizen science networks to combine with the historical information from museum specimens.
I will say that meta collections and are expanding our work to make meta collections more accessible and more diverse so that our meta collections can include the full array of types of collections. So I think that collaborative working together towards meta collecting is what I think is the next big thing, hopefully.
Well, for us in Missouri, the next big thing is digitizing the herbarium. So we just got this big donation to digitize the 6 million specimens. And we're also using the spectral data to help identify. So this will keep us busy for the next 6 years or something.
Yeah, a lot of what's on my radar and ongoing in my lab is research along the lines of what I presented today. Much of it's new, and we are expanding that research across more plant species and more regions of the world that have seen climate change more and less to really try to get a handle on what insect decline is doing to herbivory and whether there's a link. We're also trying to connect those data to the insect databases that do exist. So we have a 30-year moth data set from someone named John DeBenedictis and at the Bohart Museum at UC Davis. And one of my students, Grace Horn, is working on trying to reconcile moth biodiversity with the diversity changes that we see on the leaves of various plant species. So I think it's a really exciting time to be connecting entomological collections with these herbarium collections to even increase their value.
All right, thank you so much. Really enjoyed all of your talks and looking forward to seeing you all, everyone who registered in advance at the U.S. Botanic Garden later tonight for a reception. The doors will open at 6 o'clock. And I think, June, you have a couple of remarks to make? Hand it over to you. Thank you all so much.
Thank you very much for all the speakers for highlighting the broad contributions of collections in herbaria, natural history museums, and the botanical gardens to broad range of in biodiversity science, climate and environmental science, conservation biology, public education, as well as the history of science as a process. I think, yeah, it's really interesting to hear in the panel discussion about the new developments and the insights on collections. I think it will come from advances in technology, international collaborations of scientists across different disciplines, and our proactive dialogue with the public and the policymakers, because collections are very important, but often it got neglected. I mean, this series of discussions in today's symposium really highlighted so much we can do, especially with new technology, we can gain a lot of more insights. Thank you so much for the speakers, and also thank you for the participants for a very interactive and dynamic symposium. And as Susan said, 6 to 8 will have the reception in the U.S. Botanic Gardens. Make sure you get your tag, okay? Yeah. Thanks again. See you there. Yeah.